V. So eventually, the RNA product form that will be further will be shattered into the small pieces or the cDNA. Then the adapters are attached to the beginning and the end of each of these fragments. Like here, that is, adapters are indicated by blue and this gray color. Then this nucleotide information is amplified using PCR amplifications. Eventually, these reads are read by the sequencers. So now let's get an overview of RNA-seq pipeline, and so there is where our analysis starts. So a typical RNA-seq processing involves three major steps. First, the pre-processing, that is performed to remove adapters and PCR duplicates. The next one is mapping. It is used to identify locus of a gene as well as the distance between the genes. So mapping algorithms align your sequence read on a reference genome. The third one, that is most important again, that is quantification of gene expression level, where you quantify the expression value of your genes. So on our T-Banfo platform, all these steps are color coded. So here, for example, here the pre-processing steps are indicated by the blue color, and mapping steps are color coded with the red color, and gene quantification are color coded by the green color. So if you have any questions so far, you can post your questions in the chat. So before we look into details of all these steps, if you have any questions so far, you just post your answer in the chat box. Post your question in chat box. So if not, let me go ahead. So now let's look at the steps and the algorithms that are involved for the pre-processing. Once with the start, you are on our T-Banfo platform, when you run RNA-seq pipeline. So with start, you are actually uploading your sequence on a server. So once you start, then the pre-processing step begins. So pre-processing performed by Trimomatic and the PCR clean. So where it PCR clean removes the PCR duplicates, by Trimomatic removes the adapter sequences. Then the GTF processing. So GTF processing, as we have already told you, that GTF files are used for the annotation. So it by cup merge, it will merge the different annotation of the genome. So then the mapping. So then for the mapping, there are the different algorithms like Botai, Tophet, and HiSet2. So in the mapping, you are aligning your reads on a reference genome. Then the next one is quantification or the expression label. So where you once your reads are aligned on a reference genome, then you can quantify the expression label of your genes. So it can be done by different algorithms like Shellfish, RSCM, and HT-seq. The next one, then it's the isoform constructions. As we already we know that. So can anybody tell me what are the isoforms? So can you answer me? What are the isoforms? Isoforms, yeah, seriously. Anyone else? Apart from Hesi, anyone else know That what are the isoforms? Okay. Okay, so Shresi, you are right. Isoforms are produced by the alternating splicing. So they are two or more functionally similar but not identical sequences. So isoform construction is done by the cufflink software at this pipeline, and then the exon detection. So exon detection, what are the exons? Anybody can anybody tell me what are the exons? What are the exons? Madhuri, Shreyasi, and Neha. Yeah, you all are right that these are the coding reasons. So exon detection can be performed by an algorithm that is called Augustus. And detection of expected exons in the reference genome, this detection can be by three different approaches: live, acceptor, or the donor side, enrichment mapping, or the where they combine both the approaches. So now let's look at each step, look into the detail of each step one by one. Okay, so let's start with the pre-processing. So here, as we know that we have already during the when we have started the RNA sequencing, we have amplified our sequences using PCR, and we have also added adapter region at the start and end of the reads. So, but this PCR processing stuff what it doing? So it removing both the things, that is PCR clean. Here PCR clean removes the PCR duplicates. So the question arises why it is needed. So the presence of duplicated reads from the PCR reaction can distort estimate of the gene expression level. So the input format for this particular modules or always are the FASTQ files. So after cleaning the PCR duplicate, the we will again get the same output, that is FASTQ reads. The next is tremomatic. So this tremomatic algorithm removes both, that is technical sequences as well as the adapter sequences. So eventually you will only left with the clean sequencing reads. So eventually what we will obtain after the pre-processing. So after the pre-processing, it will clean our data, and this will increase the likelihood that the correct read must align. So these steps reduce the sequencing errors that are by mistaken can also generate for a single nucleotide polymorphism. So note, they are of course at the our platform. These pre-processing algorithms are coded with the blue color. So now let's look at how these adapters looks like. For example, here we have this particular read, and any technical sequence would be color coded like here. It could be with the pink color, and then the adapter sequences like here blue color. So this Tremomatic removes both technical sequence as well as these adapters. So next is mapping. So mapping methods are usually first they align your raw reads on a reference genome. So then they are used to identify a locus of a gene as well as the distance between different genes. So the locus means is identifying the position of a particular gene on a reference genome. So on our RNA-seq pipelines, this mapping step is actually color coded with the red color. So there are the several methods available for the mapping, and like we can either map on a full genome manually and then manually detect the alternating splicing. We can also use a known set of transcripts from the available GTF files, and then we have different algorithms like there are different algorithms like HiSet and Toffert and Botai. So now let's understand these algorithms one by one. The Botai, this Botai, it is a fast align algorithm that is based on a seed or the Kramer approach. So where seed represent the substring from the reads and their reverse complements, and these seeds or the substring and reverse complements are first extracted and then aligned to the reference genome. So in an ungapped manner, then their positions on a reference genome are recorded. That's using the GTF file. Then they are extended into the full alignment using SIMD accelerating dynamic programming. And then the next is Toffert. So the Toffert is the another fast splice junction mapper for the specifically for the short reads. So it aligns RNA-seq reads to the reference genome using Botai, which is of course a read aligner algorithm. So after the reads are aligned by the Botai, what this Toffert is doing, that it analyzes the mapping results to identify splice junctions between the exons. Then the HiSet. So it is another alignment algorithm that is alternative to Toffert or something else, Toffert 1 and Toffert 2. So it aligns reads to the reference genome and build a map of junction reads for assembling the transcripts used. So apart from these mapping algorithms, we can also use database for known transcripts that are alignment free pipeline. So then next is quantification. Once your reads are aligned on a reference genome, so why we need to do you know any algorithm for the quantification of RNA data? Do you know any algorithm that can quantify sequencing reads? RSCM. Okay. Madhuri is saying RSCM. Anyone else? Do you know how we can quantify RNA expression data? So Madhuri, you are right. So once reads are aligned against a set of reference transcript, next is quantification. So the different quantification methods like HTSEC and RSCM, they count the number of the reads against each transcript. Then it converts read count into relative gene expression label. So it can be performed by sailfish, RSCM, and HTSEC. So after the pre-processing and mapping step, these expression value of genes and the isoform are generated and displayed in the form of gene expression table. So this is how finally you can able to quantify your gene expression data. So your gene expression table can be quantified in different terms like counts. It could be FPKM, it can be RPKM, or it could be TPM. So now let's understand what are these FPKM, RPKM, or the TPM values? So depending on the input data and quantification approach that we have selected, this number can be any of them. So what is FPKM? It indicating fragments per kilobase per million. Then this RPKM indicating reads per kilobase per million. Then the TPM is similar to both of FPKM and RPKM. Only here, instead of the reads or the fragments, we are computing transcripts per kilobase per million. So how you can quantify it, as this indicated by this formula. Here first total reads are counted in a sample, then they are divided by with the million, and this million per million scaling factor. Then these divide the read counts. This normal but it will do that it normalize the sequence depth, giving you reads per million. So once you get these reads per million, then you will divide your this RPM values with the length of a gene in the kilobase. So this will ultimately produce you RPKM value. So RPKM and FPKM is very similar. The only difference is that FPKM is for the parent data. So for the with the parent data, so you will have two reads that correspond to a single fragment. So if one read in the pair did not map, the other read can correspond to the single format. While in case of the RPKM, it deals only with the single read RNA-seq data. So in that case, instead of the fragments, we will have the reads that correspond to a single fragment. So then, of course, the TPM, that is TPM is again the similar, is similar to FPK and RPKM. So apart from, so only here we are dealing with the transcripts. So if you have any question till now, you just post your question in the chat before going ahead. So if you're able to understand this so far, okay, so just before giving demo, let me just show you how our pipeline for the how it works and how we can proceed to generate a gene expression table from the RNA-seq data. So here, as I have told you, that on the TPINFO platform, all these sections and subsections are color coded by depending on the step that we are proceeding. For example, if we doing the pre-processing, this particular pre-processing step is color coded with the blue color, and then these red color steps are indicated, they are mapping steps. Then this is gene expression quantification is indicated by the green color. Then this isoform construction is indicated by the purple color. Eventually, if you want to perform any kind of analysis like differential gene expression analysis, it is indicated by the orange color. So before running this pipeline, let's just look at the data set that we are going to use in our this pipeline. So let's look at a research publication from where we have taken the data for our analysis. So in the year 2016, there was a research article that was published in the OncoTarget, where the Bradford et al. Have explored the whole transcriptomic profiling of a patient-derived xenograft models or PDX models as a tool to identify both tumor and stromal specific biomarkers. So they investigated RNA-seq as a hypothesis-free tool to generate independent tumor and stromal biomarkers and explored tumor stroma interactions by exploiting the human murine compartment specificity of the PDX or patient-derived xenografts. So in their study, authors have performed their analysis across a 79 PDX models. And they determined that most stroma can be separated into the distinct clusters where each cluster corresponds to a specific stromal cell type based on the omics profiles of the samples. So in their study, they have explored various kinds of cancers, including breast and the lung cancer subtypes. So complete publication is available at this URL if you want to understand more depth. So in our this analysis, RNA-seq analysis, we are using a data set from the PDX. So now let's understand what is this PDX or the patient-derived genogram. So these PDX models are generated when a fresh tumor is obtained from the patient, is directly implanted into the immunodeficient mice via subcutaneously or orthotopically method. So as such, these PDX models maintain the principal histological, clinical and molecular characteristics of the original patient tumor while it remains biologically stable. Since PDX models are more closely resemble and recapitulate tumor growth in humans than the standard in vitro cell line or cell line xenografts, so that's why nowadays they become a key experimental platform for any preclinical drug development. So here we will try to analyze a data set that contains a breast cancer subtypes, two breast cancer subtypes, that is ER, ER positive and triple negative. So what is this ER positive? So it is identified with the presence of a specific receptor that is called as estrogen receptor on its membrane. While the triple negative is lacks all the three common receptor on its membrane, and because of this difference, one form of breast cancer subtype is more aggressive while other is more treatable. So now let's run the RNA-seq pipeline. So every one of you have access to the T-Info platform server. So let me just share a file with you before running this pipeline. So just every one of you just download this this file where we have SVL file. So once you able to download this file, and just let me know about that. So just type your answer yes or no at the chat box once you able to log into the server, as well as able to download this SVL file, this input file. So any one of you able to log into the server and able to download this input file? Sanchita, Renuka, Sindhu. Okay, that's great. So before just running this pipeline, let me know what this file contains. This is SVL file where we are giving the URL of our FASTQ files. So if you look at this file, so here you will find there is ER positive samples and triple negative sample that I have already talked to you. And we have for each of these samples, we have FASTQ file. And for the FASTQ file, as we have already told you, these FASTQ files contain the raw reads of RNA-seq data. So we have read data for each of these files, and SVL file is only containing the URL of all of these files. So now let's move to our server. So okay, so once you are on this interface, here you will find different sections for the analysis. So as we are here, we are doing RNA-seq analysis. So we will go under this NGS data. So under the NGS data, first subsection is transcriptomics. So if you click on this first pipeline, so as this is not a demo pipeline, so this is like we are running RNA-seq pipeline directly. So if you click on this RNA-seq pipeline, you will get something like this. So if every one of you able to reach to this interface, Sriyashi. Okay, that's great. Okay, here you will find different, if we go to this file upload area, here you will find different options like type of reference genome. So as this is already, we have already, you can, if you want to use new organism, then you can upload your own reference genome file. But as already we have a human genome, so we have already have our model genome GTF. So we are selecting this model, already our model genome GTF. Then format of the NGS data. It is asking the format of your input files. As I have already shown you that SQL file is containing the FASTQ files. So where we have raw RNA-seq read data. So format of NGS data, just select the FASTQ file. Then whether it is a single end or the pair end. So in this case, it is a single end. So it could be a single end, it could be a pair end, that you can check yourself. So then this is organism. So select the organism. So here we have two, as for the human, these are Homo sapiens. So we can use the latest GTF. So here we are providing the human reference genome with the GR38 genome. So we are selecting this. So now let's go to the file uploads. Once we done with the selecting the options, now you will find here different options. So where to upload your file. So if you have a 1 or 2 file, you can upload directly. So here if you look at the bottom part of this section, here you will find upload text file or SVL file to bulk import, as we have large number of the sample. So here we will upload our SVL file. So if every one of you able to do this, so able to upload your file, once it 100% is showing it that your file got uploaded. So if you have a single file and kind of 1 or 2 files, you can even then paste your link here. Like this is SVL by selecting this. So as we don't have, so we can remove it. So as we have multiple files, that is bulk file. So that's why here we are uploading our files where we have multiple sequences. So every one of you able to go to this section. So if you next is group selection. So if you want to perform any differential gene expression analysis, in that case this step is required, otherwise you can cancel it. So if you click on this uh, group selection, you will find here your files are already got uploaded. So if you want to perform any differential gene expression analysis, then you can add the groups. So here, as I don't want to do it, I can hide both the sections. So then I can just cancel this. Cancel means I don't want to do no groups. So I will select it. So here, as we want to generate only a gene expression table for RNA-seq data from the raw reads. So just give me a second. Just let me know whether you are with me till now. Okay, Shreyas. Okay. So if you're able to upload your file and then you can select this, and then you start your pipeline. Let's begin with the start. We just going to begin our next step, that is pre-processing. As I have already told you that in pre-processing contain two major steps, that is tremomatic. So tremomatic will remove your adapters as well as any technical sequence. Then is PCR clean. So PCR clean will remove any PCR duplicates of those reads. So once we able to perform these two steps, then what was the next step is mapping. So you will have various algorithms for the mapping. So here I am performing HiSat. So this mapping will align your raw reads on a reference genome. So once we able to perform mapping, then next step is isoform construction. So here we will select the cufflinks that will generate the various isoforms. Then the cuff merge, as we have already told you, that it will merge your exons based on using the GTF file, information from the GTF file. So the next step is cuff merge. So every one of you able to reach to this section till this GTF processing, able to perform all these steps. So the next is map. Okay, that's great. So the next is it mapping on the transcripts. So these are two different algorithms that can generate isoforms, Shreyashi. So and they are using all the different mathematical algorithms. So they're performing the same thing. Isolets and couplings, they are actually used for isoform construction. The only difference is that the algorithm behind that is different. Then is the option that is getting highlighted, that is mapping on the transcript. So once you're able to identify the exons, so finally we will map it on a transcriptome genome. So this botide T, it will eventually map your reads on a transcripts. So you will able to produce your transcripts. So once you're able to map with botide T2 on a transcriptome, as here we want to generate a RNA gene expression table. So then we can perform here you can perform various different algorithms. Here the algorithm that is getting highlighted. So expression label can be quantified using sailfish, HT-seq and RSCM. So this RSCM will produce your gene expression table. So again, it is asking whether you want to generate your gene expression table in the form of RSC, FPKM, or the various other options. So here you can define whether you want to produce your outcome in form of counts, TPM, or the FPKM. I am selecting FPKM as it is a normalized value. So once we able to perform this RSCM expression, then we can end our pipeline here. So this is RNA-seq. This pipeline is also the part of as an assignment in your transcriptomics 1 course. So now so once you able to do this, then you will find the option is run pipeline. You must click on this step, otherwise your pipeline won't be, you cannot run your pipeline otherwise. So just click on run pipeline. Okay, so now how we can check that whether our this particular RNA-seq pipeline, as these FASTQ files are of very large size. It could be even a single FASTQ file is like 5 TB, 3 TB, sorry, 5 GB, 3 GB. So it takes a lot of time. It's like it will take almost 24 hours, sometimes even more than that, depending on the what are the processes are running behind at the back end of on this server. So it will take a lot of time. Here from here you can actually check with the how much your pipeline is already get processed. So here like it's only a 0%. So always it will this particular pipeline, it will take like a minimum at least 24 hours, but it could be a 48 hours sometimes, depending on the other pipelines that are running on this server. So as we cannot look into the output of this data, just I have data from the other pipeline. So that is from the demo. So just to show that what kind of output we will get. So for like here. So here you will get the so here you will get the expression in the form in form of FPKM values. So then you will have the expression in the for the genes as well for the isoforms. So let's take just one file. So after running this pipeline, you will get two kind of files. One is gene expression table, other is isoforms, and other one is not here I'm showing, as I have just run another already demo pipeline, where is not filtered. So in the not filter contain all the data for all the genes and the isoforms, even that have that have that have zero or minimum variance among the sample. So the not filtered is the only, the filtered file will contain only the gene expression value of those genes which contains where we have filtered out all those genes where the value of that particular gene is variance of the expression for that particular gene is although a zero or it is very low variance. So in filtered file usually don't contain all the genes, while not filtered file will be contain all the genes. So now this is just an example. So this kind of table you will get after running your pipeline. So it will contain the FPKM values. So let me just show you what this table representing you. So before that, just I'm asking whether every one of you able to run your pipeline. So I hope from before the next session, your pipeline will be complete and we will proceed any kind of analysis with that particular table. So here to just to show what this table representing. So Sindhu is able to complete her pipeline, so, able to run her pipeline. Anyone else? Just let me know that. Okay, it's processing because it will, I have already told you that it will take a lot of time. It could be a 24 hours minimum. So I just want to know whether you're able to run this pipeline. So let me share one table with you. So what kind of table you will get out of this pipeline? What kind of table? So let me just share that one file with you so that you can also understand yourself. So just able to complete this pipeline. If output you won't get as it will take a lot of time. So how many of you able to run this pipeline? Otherwise, I will just demonstrate it again. Just let me share you one file. So at end of running your pipeline, you will get output like this. Just let me paste it on a chat. So you can download your this file that from the link that I have provided you, or you can save this. So at end of once your pipeline will complete, it will give you output like this. So I hope before the next session, your pipeline will be complete and you will able to use your own data. So just how many of you able to complete your pipeline? Just write yes at the chat box and just download this particular file, as your pipeline will take at least 24 hours, minimum 24 hours. So then just copy your table and just paste in Excel. So you will get output like this from this data. After running your pipeline, you will get output in this FPKM values. So here, what it representing? Suresh, Benji, Samyukta, yes, okay, that's great. So here in the table, you will get the genes, and in the columns, you will get the samples. As I have already told you, our input files is containing two kind of samples. So can you tell me what were the classes of those samples? So can anybody tell me what kind of samples we are using, ER positive and triple negative? Samyukta, anyone else? Samyukta is saying ER positive and triple negative. Anyone else know what kind of the sample input, what kind of the samples we have in our input file? Okay, Samyukta, you are right. So here we have two breast cancer subtype, that is estrogen receptor positive and triple negative. So as here, if after as I have already run this pipeline before this section, because it will take a lot of time. So before the next session, your pipeline will be completed and you will get the output like this. So here the columns are representing the samples while rows are representing the genes. So what is this ENC or G? So it indicating the ensemble ID of these genes. So these are the samples, ER positive, and then we have samples that are triple negative. So now you can perform any kind of analysis on this data once you able to obtain this particular structures data in form of a table. So if you just look at the bottom of this table, so here you will find the ensemble IDs like this. That is ENS MUS. Can anybody tell me what it indicating? Can anybody tell me that what it indicating? That is, let me just make a size bigger. So what these IDs indicating? ENS MUS, but it represent. Can anybody tell me that? So if you don't know, human city says no. Madhuri, human space. So ensemble ID, yes, it representing ensemble ID. So but this ENS represent the ensemble ID. So then the next is MUS. So what it indicating? Anirudh, yes, you are right. It indicating this particular, these genes are belong to the mouse, that is mus musculus. So ensemble IDs, that are the genes that are specific for the mice. So those IDs are represented with ENS MUS. So and beside, if you're looking at the human gene, so just let me highlight it here again. So what it is written? ENSG. So G indicating, if this is only till this particular ENSG, it represents this gene belong to the human. And ENSMUSG representing the gene belong to the mus musculus. And then if instead of this G, it's like it is T. So what it represent? So in that case, it representing this particular ensemble ID belongs to a transcript. If it is G, then it indicating that this particular ensemble ID is associated with the gene. So you must able to differentiate. So, and then different genes you can search, and you on a Google even a particular ID, so which gene is correspond to that. So every one of you able to understand it. Just write your answer, post your answer in the chat. So eventually at running that pipeline, you will able to obtain this particular gene expression table. So Anirudh, yes. Okay, Sreshi, everyone. So far what we have discussed, that how we can pre-process our data from raw reads to this structures table. So on once we have this data, now we can perform any kind of analysis. Just for example, let me show you just a simple how we can differentiate. So So let me just select this particular gene. And let's see how we can visualize this gene and we can perform any kind of analysis. So now if you are with me able to do this, just plot a column. So if we looking at this particular gene, this is how we can analyze it. I have not performed any kind of normalization so far. That we will discuss in the next section. Next session. Here I'm just showing, once you're able to obtain a gene expression table, you can perform any kind of analysis. For example, if we're looking at this particular gene, so these are different samples, and we can look at here at this bar plot that this particular gene is actually expressed only the 3 samples, out of which 2 belongs to the triple negative, while 1 belongs to the ER positive. So this is how, once you obtain your gene expression table, you can perform and understand what this data representing. So are you able to download the data, gene expression table that I have provided, URL of which I have provided you? So for example, now let's just look at, I think you might have, suppose I just want to search one gene like KRAS in sample ID. Let's see whether this particular gene is available in our samples or not. So now let's go over our table. Let's go over in our table and search whether this gene is present or not. So this gene is not present. Let me search some other gene like so now let's search this particular gene. This is also not available. Okay, so like we have a gene that is ACTB. So if you search with this ID for any gene, so then you will get what this particular gene, like once I search on this ensemble, I found this particular gene is CDKNC, and then, sorry, this is okay. So this is just unable to search this. Okay, gene card. Okay, so in gene card, we can search this particular gene. If you click on this, so then you will get detail of this particular gene. So ensemble ID of this particular this, and this gene is ACTB gene. So it is actin beta. So the detail of gene you can find in the gene card as well in the ensemble. Right now, just ensemble is not working right now. Maybe some network error or something like that. So then you can take this gene and analyze how it is getting expressed. Okay, so let me just paste this gene here and IDs. So here. So now let's see how it is expressed between different samples. So now let's select any gene with the this ID with the sample name with sample IDs, and just see look at the expression label. So just insert and plot a bar plot. So now you can look at this particular gene which was ACTB with represented with this ensemble ID. So how it is expressed in different kind of samples. So are you people able to do this? You can visualize the expression, your gene expression in different samples. So you can just do this, only you can plot just a bar plot, and you can look at this, how this gene is expressed in this ER positive samples, and then these are the triple negative samples. So this is how you, once you get your gene expression table, you can perform various kind of analysis, and you can look that how it is significantly differentially expressed in different kind of samples. So these kind of analysis you can perform once you have this gene expression table data. So now let's go over back to our presentation. So by running this pipeline from the FASTQ files, we able to obtain a gene expression table which can in the columns where we have a samples, while in the row they represent the genes. So in each of this cell it represent the value of this particular gene in this particular sample. So in each cell you can visualize the expression label, or you can quantify, able to understand the gene expression label of this particular gene in this particular sample. So now all the topics that we have discussed today are covered and very well explained in the transcriptomics one. So please complete this course before the next session, so that you will understand what we are going to why we are doing all these steps, and you will understand, you will get the description regarding all of these steps and the algorithms. So before the next session, so some of you who have yet not completed your profile, so before the next session, complete your profile and complete the course transcriptomics one. I hope you most of you must have completed introduction to the bioinformatics, introduction to the bioinformatics till now, and try to run this RNA-seq pipeline. So let's and if before the next session, so just visualize your results. So before performing any kind of analysis. So the code, and I hope every one of you before the next session would have the access to the coding platform, and where I will demonstrate you. Just explore the coding platform before the next session, and in the next session we will see how we can visualize and analyze our gene expression data once we already obtained this gene expression table. So I will demonstrate you. So just explore the coding platform. So I think this is for today. So Mohit sir, if you want to add anything.
Thank you, Harpreet. And I just wanted to ask everyone to complete the things that we discuss. And soon we will be discussing about coding in much more depth. And after we learn about the NGS technologies that we are covering in the first week, the second week, and the third and fourth week will be more about the technology application. So it's better to be prepared and at least finish the courses and have this entire access ready. So you should have access to all the platforms. You should be aware of where is, so that we can guide you and you can directly go there and see it. Like today, I think everyone did really well, downloading the resources, running the pipelines, and analyzing the output. It's a great start, and I think if you keep on practicing some of it that we have done today, and you will also receive the recording in a while and communicate with other students who are in your group to help them finish these simple tasks. So with this, I will end this session, and we will meet again next week, Monday. And for now, you all have a nice weekend, and let us know in the forum and everywhere about the problems or any issues that you face, and do practice during the time that you have. And we will discuss progress on Monday as well. Have a nice weekend. Bye bye.
So if before ending the session, I just want to say one point. I just want to add one
Sure,
point.
sure, Harpreet, please go ahead.
Okay. Okay, just let me share my screen. So please, on the forum, if you face any problem during your course or running any pipeline, you can ask your questions even at the forum. As here I'm just showing the Sanchita already posted her problem. So here we have also tried to address her. So if you face any problem, post at the forum. So any question do you have, and are you facing problem during running a pipeline or going through the course, you can post here. Like already, I think Sanchita posted. So you can do this as well. Thank you. So thank you everyone. We will see you.